This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 237, recorded on March 4th, 2021. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Hello there. Nice to be with you.
Hello. Nice to see you again, Dr. Schaechter. Hope all is well out there in the sunny state of California.
Absolutely.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello. Good to see you all.
Now, Michele, have you played any golf yet this year?
No, we still have snow, Vincent, but thank you for that thought.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
How you doing? Is Charleston good?
Charleston is good. On Monday, we had close to 85 degrees, and it was truly a beautiful spring-like day, and then winter returned on Tuesday.
Well, this is roughly the 10th anniversary of TWiM. We hit 10 years last month, February, so pretty close.
Yep.
So we decided to do a little something different for you. So stay tuned. Don't turn it off, please. Who used to say, don't touch that dial? There was someone who said that. Anyway, we started, our first episode was February 23rd, 2011. So that's 10 years ago. And for the first episodes, we had a mix of hosts. Michael Schmidt was there from the start. Sometimes we had Margaret McFall-Nye, sometimes Joe Handlesman until she became an advisor, science advisor at the White House. Sometimes Cliff Mintz, sometimes Stan Malloy, Ron Atlas, and many others came and went. And towards the end of the year, Elio joined us, and he became a regular. I guess he can't keep away because he's been here ever since.
No way. I'm incapacitated.
He's incapacitated.
And then Michele joined us in 2013 after she had been on an episode at an ASM meeting.
That's right. I think that was my audition. I didn't realize that I'd have an opportunity to join this august group.
And you said to me, I'd be happy to help out when Elio's not available. Okay.
Yeah.
And then it just morphed into being here every episode, which we greatly appreciate, Michele.
My pleasure.
And since then, it's really been the four of us. Sometimes not all four of us are here, but it's quite a run. It's really impressive that all of you are willing to do this. I surely appreciate it.
We're privileged. We're privileged to do it with you.
Oh, thank you so much. I don't take it for granted, that's for sure. But I just enjoy it so much. I was going back, looking over the old episodes. So much good science we talked about. It's amazing, and even some interesting titles to the episodes.
Yes.
So 10 years. That's and we've got this is our 237th podcast. So we're a little bit behind TWiM. But that
we need a bacterial pandemic.
No, we don't.
Really.
We do not.
That's a good question, Michael. When's the last bacterial pandemic that you can remember?
It's probably plague.
Right?
Yeah, it's the pneumonic form of plague that we had in the Middle Ages.
Yeah, it's a long time ago. Why haven't we had plague bacterial pandemics? Because we have antimicrobials?
In the modern era, certainly.
Yeah.
But I think the whole issue of bacterial pandemics is it brings the host down so quickly, unlike the viruses like we're seeing with SARS, where you can have a substantial component of the viremia that is occult, where the patient is not aware that they are infected and literally transmitting the virus. With the bacterial infection, especially the pneumonic form of plague, people went down very, very quickly.
And some have suggested it's because bacteriologists are just a little bit more clever than virologists, but I don't think that way.
It's a very interesting thought, yeah.
Oh, we're going to get letters on that for sure.
But, you have to admit, Michele, in the last year, the virologists were pretty clever. They came up
Oh
with
my goodness. Yeah.
vaccines in no time.
Amazing.
So, go ahead, Mike.
I was just going to say it's what the molecular biologists, the virologists, and let's not forget the folks who have been responsible for the chemical synthesis of these vaccine preps. They have done truly a remarkable job in addressing this, and hopefully we'll begin to see the crash of the pandemic curve as we begin to vaccinate the globe.
And I've got to put in a plug for the immunologists out there as well.
Yeah, we thank them all. So TWiM at 237. That's number 2 in the microbe TV stable, right? It's right after TWiV, and it's in front of This Week in Parasitism. Now, TWiM is twice a month typically, and all the others are pretty much once a month. So that's what explains the numbers. But I hope we get another 10 years out of this. That would be nice.
Yep.
I certainly will continue to do it as long as I can because I find it enjoyable. And I will do it long beyond the time I am here at Columbia. I don't know how long that will be, but I'll do this as long as I can.
That's good to hear. Thank you. To hear.
I mean, Elio, you're very familiar with retiring from academics and continuing to do work in the field, right?
I'm well acquainted with ancianity.
So I will do the same. Anyway, for this episode, we asked a number of our colleagues, all of who have been on one episode or another of TWiM. We asked them a question. We said, please give us 5 minutes, record your thoughts on how our discipline has progressed, changed, contributed to the advancement of our understanding of the microbial and macro world in which we live. It's a tall order, of course, but with 5 minutes, you have to restrict yourself. So I'm going to play these for you.
We have quite a bunch, don't we?
Yeah, we have a good response, although not everyone agreed to do it. We do have a lovely group of individuals. And so let's start. I will play them. So the first is Jennifer Baumberger from the University of Pittsburgh.
Hi, TWiM. This is Jen Bomberger from the University of Pittsburgh. I'm an associate professor in the Department of Microbiology and Molecular Genetics. I want to thank Vincent for the opportunity to send along my perspectives and excitement for where I see our field going in the coming years. My laboratory studies microbe-microbe interactions in the respiratory tract, and I joined TWiM a few years back to talk about some of our studies on viral bacterial co-infections in the airways of patients with cystic fibrosis. Mm-hmm. One area of microbiology that I've been closely following and enthusiastically watching develop in recent years is our evolving understanding of polymicrobial infections and polymicrobial interactions during disease. In this era of next-generation sequencing, we're now aware that polymicrobial interactions in disease are happening much more commonly than previously known and could be thought now to be the rule rather than the exception. There are excellent studies demonstrating that bacterial interspecies interactions can change antimicrobial susceptibility profiles, which shapes how we think about clinical microbiology in addition to microbial pathogenesis research during disease. We also know that microbe-microbe interactions can shape the evolution of a microbe in its environment and greatly change disease progression. We see fascinating examples of this in the cystic fibrosis microbiology field where I work. There are also many excellent studies examining viral bacterial co-infections, where the host mounts an immune response to one pathogen, and this can then leave an Achilles heel to a second invading microbe and allow secondary infections to develop. We see the devastating effects of these superinfections during seasonal and pandemic influenza cases, and even now during these current times during the COVID-19 pandemic with infections with SARS-CoV-2. All this to say that I'm looking forward to our field progressing in our understanding of how microbe-microbe interactions impact human disease. My lab, and I know many others, are striving for more sophisticated models to study polymicrobial interactions, particularly in vitro models to allow for mechanistic studies, as well as continuing to develop our ability to translate observations in the microbiome to mechanistic studies of microbe-microbe interactions during disease. Thanks so much for letting me offer my 2 cents, and I look forward to hearing the other perspectives being offered. Hope you're all staying safe and healthy.
Lovely.
Nice.
Lovely.
Lovely. Jennifer was on TWiM 141, where she talked about the work of her lab on how a respiratory virus enhances bacterial growth by dysregulating nutritional immunity. And I had visited Pitt and spoken with Jennifer one-on-one, and I said, this would be a great story for TWiM. So she said, sure, anytime, which I love to hear.
And you signed her up.
Signed her up.
Then and there.
And she's obviously quite eloquent, and look forward to it. Yeah, I think this polymicrobial story is understudied and underappreciated. What do you guys think?
This is exactly where medicine is going. Polymicrobial infections, especially with the advent of inexpensive next-gen sequencing in the clinical laboratory, really highlights the growth potential that we are going to see in our staff, where we're going to need a bioinformatician to be able to first reduce the data set that comes out of the NGS devices, as well as the infectious disease physician who is going to basically look at all of the microbes associated with the particular infected area that the specimen came back from and interpret whether or not the microbes that are resident in the specimen really warrant concern. And then at the same time, because it's NGS, the bioinformatician will be able to probe that specimen to determine the resistome of the community that is resident in that patient's specimen. And then the infectious disease practitioners are going to have to make an informed choice about whether or not an antimicrobial is warranted, because NGS won't give us susceptibilities, but they will tell us about the potential antimicrobial susceptibilities of the community. So in essence, what Jennifer was telling us, it's microbial ecology, folks, and we have to understand what's going on. So if you listen to our back catalog, you'll get really a sense of where our discipline is been and where it needs to go if we're going to continue winning the war of fighting infectious diseases in the human host.
And if I could add, not only are we bringing together different microbe types to study them and understand the impact, but also we need to bring in the infectious disease doc, the microbiologist, the computational biologist. So our field is getting much more scientific as well.
Yeah, that's good.
Nice.
All right, our next comment is from Joe Handlesman, who originally spent maybe a year on TWiM.
Yeah.
She was at She was at Yale University at the time, and then she went to the Office of Science and Technology Policy at the White House that was during the Obama administration, to advise on science, and then now is at the University of Wisconsin. So let's hear from Joe.
It's been an exciting decade for microbiology. Several big advances have changed my part of the field, microbial ecology. During the last 10 years, we moved past the sequencing frenzy, which involved just sequencing everything in sight, 16S genes or metagenomes, and we progressed toward a much more precise approach where sequencing had more purpose. It was done in designed experiments in which variables were manipulated, and therefore mechanisms could be studied and identified. This decade also saw the dawn of what I call metagenetics, the use of mutant analysis within microbial communities to identify characteristics that determine either the behavior of the communities or of the individual species within them. I think this is equivalent to when we first started as a field using mutants to understand how bacterial cells function, except now the unit of study is the community.
Any thoughts on that?
Joe has dropped the gauntlet. You know, this whole field of metagenetics we're witnessing it happening in real time with the emergence of the SARS-CoV-2 variants. We're seeing natural selection taking place as the virus begins to change its spots. And we have to appreciate that this is going on continuously in the microbial world with our microbiome, especially when we, change our nutritional status, we introduce antimicrobials, and we really need good model systems in order to be able to do, as Joe said, the one variable change at a time to really get towards mechanism. And on previous TWiMs, we've looked at a lot of different mechanisms of how microbes get away with doing things. And Elio's brought up many of those by introducing us to the symbiotes, which really help us dissect the way microbes interact with their local environment.
Yeah, and Joe's comments also emphasized how when there's an advance in some other part of science, in this case, computational biology and sequencing, it opens up new questions and an amazing new window into the microbial world. And I think that a parallel was when I was coming up in my training, and fluorescence microscopy became more accessible. It allowed us to study not just the bug in pure culture, but the bug's interaction with the host cell. And that really gave birth to cellular microbiology. So I'm excited about the new windows that have been opened into the world and look forward to what's next.
So I do. Joe was on quite a few episodes, but I think she returned for 100. She had already been at the White House when we had 100, and she came back and talked about some paper. And I remember she couldn't tell us anything about what was going on at the White House, either before or doing. She was very secretive about it. Very interesting.
Oh.
Although afterwards, when she left, she did tell stories. And in fact, at an ASM meeting, I remember her giving a talk about wonderful interactions with Obama. It was really good. Really nice.
The one thing that comes out of this is that the training of young microbiologists has to include a lot of new stuff. It has to include a lot of bioinformatics, for one thing, and the ability to put together all these various disciplines. So it's going to be a different kind of a training.
I agree with you. However, I have to say, here in my microbiology and immunology department, there is no such training. If you end up in a lab that has that requirement, you will get training from the lab. But I think there should be universal training so everyone understands how to build a phylogenetic tree, how to do searches.
They better, otherwise you're not going to be able to use the tools that are available now.
Well, I can share that there is a national volunteer group called Software Carpentry that Pat Schloss, my colleague here at Michigan, is engaged in. He and his trainees give workshops on kind of the basics of applying computational skills to analyzing data sets.
That's good. All right. Our next guest, I suppose, is no stranger to Michele. He's Harry Mobley from the University of Michigan. And Harry was on episode 114, which we actually recorded in Ann Arbor. The occasion was a milestones celebration where the site was designated a milestone by ASM. And so on that particular TWiM, we had Harry and also Michele and Elio. You were there as well.
Yeah, I think so.
Yeah. Milestones in Blue. Yeah. Perfect name. All right, let's hear what Harry. Now, Michele, he used to be the chair of the department, right?
That's right. And we have a new chair, and he is focusing his attention on his lab.
Okay.
My name is Harry Mobley, and I'm a professor and bacteriologist at the University of Michigan in the Department of Microbiology and Immunology. I was asked by my TWiM colleagues to give perspectives on the advances that our colleagues have made as technology developed in our field of bacterial pathogenesis. Thanks for the opportunity to reminisce. First, I recently finished autobiographies by heroes of mine, Stanley Falco and Rita Caldwell. Stan founded, in my view, the field of molecular pathogenesis, and Rita championed women in science and organized efforts to push big science forward at NSF, for example, and linked microbial ecology with the spread of infectious diseases such as cholera. These were two leaders among many that I admired in the generation that preceded me. I opened my lab in the infectious disease division at the University of Maryland School of Medicine in 1984. In this era, we had the use of only a few restriction enzymes and could isolate plasmids on cesium chloride gradients using an ultracentrifuge. We could make cosmid gene banks with inserts of 48 kilobases of bacterial DNA and screen these clones for phenotypes. We could make transposon libraries and do southern blots. And cloning of virulence genes was tedious, and as was the construction of deletion mutants. For context, at that time, there was no internet, no email, no mobile phone, no PCR, but we did have 4 TV channels. We had classical bacterial genetic techniques, and sequencing of modest lengths of DNA was achievable in the lab, but otherwise we had the biochemical techniques of generations of scientists. We drew our own figures on a drafting table, and everyone submitted their manuscripts by U.S. mail to the journals. Being in Baltimore, we drove our NIH grant applications to D.C. the day before they were due and dropped it off at the loading dock. Nevertheless, this was a pretty exciting time to get going in our field. Most mechanisms of bacterial virulence were not understood, and that's an understatement. Some of the things we did know were that Vibrio cholera and enterotoxigenic E. coli secreted diarrheagenic toxins. Uropathogenic E. coli could stick to uroepithelial cells using adhesive fimbria that was later called P. fimbria. Pseudomonas secreted a bunch of degradative enzymes, and enteropathogenic E. coli caused diarrhea in children. Staph aureus caused abscesses. But how did they cause disease? I can think of a few remarkable examples worth noting. We knew that bacteria used the Seq system along with cutting a signal sequence to secrete proteins from the bacterium. That was one type of secretion, and we didn't know any more. Fellow scientists went on to characterize at least 7 more mechanisms of secretion, including type 2, type 3, type 4, autotransporters, type 6, and so on. Elucidation of the type 3 secretion system was perhaps the most remarkable, explaining some of the pathogenesis of enteropathogenic E. coli, Salmonella, Shigella, just to name a few. Overall, the study of bacterial virulence gene expression has made the most impressive journey. Our colleagues first looked at expression of a single gene in culture medium, then in relevant media such as in tissue culture, urine, serum, bronchial alveolar fluid, and then in animal models, and then in human host tissue. Then we advanced to measuring gene expression during naturally occurring human infections in simple systems, including urine collected from women with active UTIs, or in rice water stool from cholera patients. Next, fellow scientists are tackling pathogen Rna-Seq in complex organ systems where host RNA drowns out bacterial RNA. I think this is the holy grail of sorts, measuring bacterial gene expression right where the infection is raging, not just in broth in a test tube. Our community of microbiologists will continue to sort this out. A relatively recent breakthrough has been the measuring of growth rates in vivo by determining peak to trough ratio of the origin of replication to the terminus of replication, which correlates with growth rate. Thus, doubling time can be determined during infection of the host. Finally, over the years, large-scale screening of virulence and fitness genes was advanced by remarkable cleverness, including IVET, in vivo expression technology, IVIAT, in vivo induced antigen technology, STM, signature tag mutagenesis, and now TNSeq with the ability to simultaneously assess the global contribution of pools of hundreds of thousands of mutants in cases where there are no infection bottlenecks. It's hard to imagine new advances over the next decade that will mark this level of advancement, but they will come, I have no doubt. To close, I want to thank Vincent and all the TWiM crew for their fabulous service to microbiology community, for providing more than a decade of public dialogue about our field. It is truly remarkable.
Thank you, Harry. That was lovely.
Yeah.
I think that for me, bacterial pathogenesis has been an amazing advance in the last 10 years compared to what we used to do, as Harry pointed out, and the limitations and the techniques. And that's one of the things I love on TWiM is to learn, the battle for iron and the competition between microbes and how it drives pathogenesis.
Right. Yeah, the problems are age old, like battle for iron. But now that the techniques that we can bring to bear to understand how this is happening, not just between the iron molecule and the microbe, but actually within an environment, within a community.
And then those old techniques that he measured, I had deja vu when he said cesium chloride gradients. I still remember the first time I took a needle and with the UV light was looking at the ethidium bromide band, trying to aspirate out where the plasmid had banded in that cesium chloride gradient, trying not to contaminate myself with the mutagen, because it was before the age of gloves.
Yeah, we all used to use ethidium bromide, and I didn't wear gloves.
No,
Wow.
we never did.
But
Oh,
then
my
they found out that it's not actually a mutagen for people, so we're all good.
Yeah,
We're all
but it's
good.
that P32 that we use that's, a little bit spooky.
Elio, does pathogenesis resonate with you?
Of course. Pathogenesis has been part of microbiology from the very beginning. Okay, and the fact that it was simple and primitive and not very exciting in a way because the tools weren't there, that doesn't take away the fact that it was in the forefront. I mean, Pasteur and Koch were pathogenic microbiologists.
Right.
And a real driver for the field,
Sure.
motivation to understand the basis of infection.
The interesting part is, Koch was a doctor and very much involved and very much oriented towards pathogenesis. But Pasteur was a chemist, and he dealt originally a lot with bacteria in nature. And so the fact that he became interested in pathogenesis is kind of remarkable. And he, of course, did phenomenal things. I mean, he created the first on-purpose made vaccine with anthrax and stuff like that. So, it was interesting that he took over pathogenesis. It's interesting.
Yeah. All right. Our next guest is no stranger to TWiM. It's our good friend Mark Owen Martin from the University of Puget Sound.
Oh my goodness!
And he Mark has been on TWiM a number of times, and he's always a pleasure. And so
he is.
It was not no trouble whatsoever to get Mark to comment.
Hello, you wonderful people at This Week in Microbiology. Congratulations on 10 years of bringing matters microbial to the outside world via this wonderful podcast. I want to give a definite shout out to Vincent Racaniello for actually putting this together, and not just TWiM, but all the others that I tend to call TWIX, with the X being whatever else that the podcast might be about. Elio Schaechter, who is just an invaluable and enthusiastic role model for everyone. Small Things Considered is one of my go-tos. Michele Swanson is unfailingly positive and insightful. And then, of course, there's Michael Schmidt, who suffers from the same kind of liking to get enthusiastic and talk a lot issue that I deal with on a daily basis. So it's a wonderful group of people explaining wonderful things about microbiology to the wider public. And I cannot emphasize how important that's been to me. And I'd have to say it's in three areas: the professional, the personal, and the pedagogical. And as far as professionally, it is an absolutely invaluable way of looking at really interesting, cutting-edge types of research, explained bit by bit. Now, yes, it's true that there's banter and back and forths about things, because when folks get to know one another, that's going to happen. And you'll see a lot of that in podcasts, so that doesn't bother me in the least. What has been so important to me is that if I have difficulty understanding a paper, and that paper is covered by TWiM, I have absolute confidence that it's going to help me. In addition, I've been fortunate enough to have been a guest on TWiM several times, and it's meant so much to me, giving me kind of an outlet for the kinds of things that I need to talk about, especially when it came to a TWiM that I did with Vincent a few years ago about papers for introductory microbiology students. I got so much wonderful feedback on that. As far as personal, I think that many of us, especially at small liberal arts colleges, are the only microbiologists that's present. And because we're the only one there, we don't have that quality quorum around us. And This Week in Microbiology provides that to me. I know there'll be other people that share my enthusiasm with matters microbial, and I cannot emphasize how important that's been. In addition, I have had wonderful interactions with every single member of the staff involved with this. I mean, Elio has always been fantastic. Vincent's become a friend. Michele is so patient with my eccentricities, and Michael is just so much fun to listen to. So I felt like I was included just because I'm a little bit part of it, but also because I've gotten to know the people. And it really helps me personally because I often feel isolated because I'm not in a place with a lot of microbiologists with whom to talk. But pedagogically, I have to say how important what you do is. You explain microbiology. Now, some might say it's being pitched at a level that the public doesn't really want to delve into, but this isn't true. But remember, from number one, it's not true, but I would also like to emphasize that it works really well as an educational tool. And I have been using it as I've been using Small Things Considered with my own microbiology classes and also for my introductory classes in cell and molecular biology. So what I'm trying to say as I hit the 4-minute mark is that this podcast matters. And I believe that I'm not the only person willing to say that. I know many, many people in the microbiological community that feel this way. And the most important thing to leave you all with is that you've done a really wonderful job in helping to promote a quality quorum of other microbial enthusiasts. And I want to thank each and every one of you, Vincent, Elio, Michele, and Michael. Congratulations on 10 years, and may you have many more. Thank you again.
Wow. Lovely words.
Very nice. Thank you.
We really
thank you
have
for
to
that,
thank
Mark.
Yeah.
We actually weren't looking for praise of TWiM, but we will take it, no?
It's interesting that he points out the niche that this fills. We didn't plan that. We didn't think that this would be a way to establish a community of microbiology or people interested in microbiology. And yet it worked out that way. So there is a byproduct here, which turns out to be a very important, almost central part of what we do. Interesting.
And if I could make a pitch for the podcast that Mark was first on with Vincent, namely, what papers should an undergraduate read? It's still pretty fresh. It hasn't aged out. And as you begin to move into microbiology, whether you're an undergraduate or a graduate student, some of those key seminal papers, maybe we could go back and revisit what papers would someone new to the field, what papers should someone begin to read in order to get a sense of where microbiology has been and where it's going and where it needs to go. And I think that's been some of the themes that we have heard just listening to these perspectives of the need for models, understanding bioinformatics in the context of microbiology, and really putting it all together. And Mark sort of sums that up with his catchphrase of the quality quorum. You need this breadth of people with experiences in order to really push our science forward.
And of course, when Mark comes to podcasts, he always brings toys. So when we've had him on at ASM meetings, he brings tardigrades and other things.
Buttons.
He makes buttons for his classes. He sent me many 3D printed viruses. So it's all wonderful. Give a little levity, which I think is really important, right?
Sharing his enthusiasm. And it has meant so much because as much as the four of us enjoy getting together and talking science, it's really learning and appreciating that people are out there listening and it makes a difference that inspires us. It certainly, every time I hear from somebody referring to some discussion that we had, it just motivates me. I want to up my game and do a better job. And certainly, Mark, you've set such a high standard for the professionalism and the enthusiasm that you bring to your classroom, that, I want to do a better job. I want to try to live up to the standards that you set. So thank you very much for your kind and encouraging words.
All right. Our next guest is Robin Patel, who was on TWiM 150 That was an episode in recognition of National Medical Laboratory Professionals Week, and she told us about directing a clinical bacteriology laboratory. So let's hear from Robin, of course, is at the Mayo Clinic. Let's hear from Robin.
Hello, Vince, Mike, Elio, and Michele. Thanks for inviting me back. It's exciting to be here. Over the past year, there have been so many changes in the field of microbiology, and I really appreciate the opportunity to comment on a little bit of what I've observed. I think we're all really excited about vaccines right now. I personally just recently got my COVID-19 vaccine, and I was never so happy to get a vaccine as I was with this one. But I don't want to talk about vaccines today. As you know, I spend most of my time working on diagnostics. Diagnostics were a focus very early on in the pandemic. And I would say, sometimes now we take them for granted, which I think is a good place to be. But I also want to recognize that there have been huge advances in diagnostics. First, public health and clinical microbiology laboratories rapidly brought us the diagnostics that we needed for SARS-CoV-2. And then industry, companies, and the government stepped up to bring us the diagnostics that we need for this pandemic. But in addition to that, there's been a lot of advance that has happened as well. I don't know if you remember, but back in 2016, the American Academy of Microbiology convened a colloquium to examine point-of-care microbiology testing with participants in the colloquium, including representatives from microbiology laboratories, industry, and the government. And they together made recommendations regarding the implementation, oversight, and evaluation of point-of-care microbiology testing. Well, a lot of this has really come to fruition with the pandemic, and I hope that we continue to ride the wave of innovation that's come with this pandemic for other infectious diseases testing. My colleague at Mayo Clinic, Dr. Brad Karen, and I wrote a commentary on the colloquium report that was published in the Journal of Clinical Microbiology back in 2017, 4 years ago. And it's been interesting to look back on what we wrote about then in the context of what has happened now. So what have we seen happen that's so exciting? Well, first of all, so many companies interested in developing microbiology diagnostics and getting them out there. Diagnostics that are easier to use than what we've had before, that are faster, that can be done at the point of care, including at home. We've seen advances in terms of self-collection of specimens. I've always been excited about this. We did studies on this that showed that people could collect their own specimens if they wanted to. But this has been slow to be adopted into modern medicine. And thanks to COVID-19, we now know that this is completely possible and actually makes a lot of sense. I know a lot of people really welcome the opportunity to collect their own specimen for a whole number of reasons. We've also seen innovations in specimen types. I don't think that Dr. Karen and I really thought about that when we wrote our commentary, but the use of saliva, for example, is particularly interesting. And I can only hope that as we exit the pandemic, and hopefully we're headed in that direction, fingers crossed, that we continue to leverage what we've learned and the technology advances to solve other problems that we have in medicine, in infectious diseases, with advanced diagnostics and use of advanced diagnostics. We knew we needed redesign of workflow of how these tests are deployed and patient care processes, and that we've seen happen. We knew we needed self-collection, and that we've seen happen as a result of the pandemic. And we knew there were distinct benefits to some of the advanced diagnostics in unique healthcare settings, such as smaller healthcare settings, including in the rural parts of the United States and developing nations, as well as convenience clinics, and as I mentioned, at home. And we've seen all of that happen with this pandemic. So thanks so much for letting me comment a little bit on what we've seen happen with diagnostics. I'm always super excited to talk about them. And I hope we continue to see innovations in this area into the future. Thanks, and have a great day.
I believe that Robin makes a very important point. For most of my career, the world of basic microbiology and of diagnostic microbiology were totally distinct, separate, and no way would they talk to each other. And now this has changed completely because, in part, the technology which is used in diagnostic microbiology is the same technology that is used in molecular microbiology. It's the same thing. So people share the same, by sharing the same technique, they share a common language. I think it's a wonderful development.
It is. And I've been fortunate to train in my lab 3 MD PhD students who have gone on to being physician scientists, but their focus is on the clinical microlab, and they do their research as well as really pushing the techniques forward. So it emphasizes your point, Elio, that there's just such a overlap between the research lab and the clinical lab and healthcare.
Yeah.
I think Robin hit the nail on the head with specimen type and self-collection, because in order to really put us on the road to recovery, we're going to need more frequent, not less frequent testing. And I think one of the early notions that came out of our sister podcast, TWiM, is the notion of a lick-a-stick test, where you would effectively lick a stick in order to determine whether or not you were contagious. And we can well imagine how moving future, as our globe gets more populated with time, we're going to be needing real-time diagnostics that people can perform without having the need to seek specific specimen. And again, looking at saliva, looking at rapid diagnostics that can afford a determination whether or not you represent a risk to others, and then your own personal behavior to stay put if you are indeed a risk.
I remember visiting the clinical micro lab here in 2019, and I was blown away because I don't go into these labs, right? I hadn't been in one since I was a grad student. And I mean, mass spec, PCR machines, huge multiplex PCR machines, amazing robotics, completely different. And this is really an area that's seen amazing changes, I think.
And I'd love to see us marry that with the growth in like online shopping, where you can have food or, any number of items delivered to your doorstep after some keystrokes. And if we could apply that same infrastructure to make it easier for people who are homebound, whether it's because they have a small child or they're elderly and don't drive, where they can have a rapid test and then get the results and know whether or not they need to see a physician, would be fantastic.
All right. Our next guest is Stanley Malloy from San Diego State University, and he's been on TWiM any number of times. So let's hear from Stanley.
I had the pleasure of being part of the TWiM team in the very first episode in February 2011. We talked about 2 manuscripts that day. The first paper we talked about presented evidence that a DNA sequence in Neisseria came into that bacteria from humans. This was a great example of an exciting new finding that was unique when we talked about it 10 years ago. But now we know of many other examples like that. The second paper we talked about was about the ability of copper to kill bacteria on contact. This is still a very important research topic today. In fact, about 2 weeks ago, Michael Schmidt was interviewed by the Washington Post about the scientific basis for using copper as an antimicrobial. I think these 2 topics nicely exemplify why TWiM is such fun. In addition to hearing Vincent, Elio, Michele, and Michael talk back and forth about the weather around the U.S. and other puns, It's a terrific way to learn about some cool science that sometimes changes the way we think and other times ponders an old scientific problem that's constantly evolving. Way back in 2006, well before the concept of TWiM infected Vincent's neurons, Elio and I wrote a paper about the impact and future of microbiology. We emphasized the importance of microbiology as a foundational science influencing all other areas of science. And we made a set of predictions about future hot topics in microbiology. An endeavor that requires a substantial amount of chutzpah. But in fact, we've seen each of the predictions that we made propel the field of microbiology forward over the last decade. And all of those predictions have been part of the TWiM conversation. For example, we argued that even fields like psychology would be influenced by our understanding of the human microbiome, something that's been proven over and over again, although there are still many questions about what, why, and how. We argued that new approaches like metagenomics would provide insight into fundamental biological processes in fields ranging from medicine to ecology. But understanding those insights would require a basic knowledge of cell biology, metabolism, and genetics, the fundamentals of basic life sciences. A great example of this is antibiotic resistance. Antibiotic resistance has been a public health concern since way before most of the listeners of TWiM were born. Mm-hmm. Although we know now some things that we can do to decrease the selection for antibiotic resistance, I'm convinced that this isn't a solvable problem. It's going to require a constant effort to develop new innovative antimicrobials to deal with the evolution of resistance. On the other hand, the new experimental approaches that have helped us understand microbiology and biology better have also provided identification of the genetics of antibiotic resistance and numerous vulnerable targets in the microbes that will provide great fodder for future next generation antibiotics. All of this is well and good, but the other thing that's really essential, if you're going to take these scientific ideas and have an impact on our society, we have to have a better public understanding of science. How can you get people to follow scientific advice if they don't know the difference between science and pseudoscience? And why would people support the government investment in scientific research if they didn't appreciate the value of science? Listening to the conversations on TWiM, the context of the problem described in the papers, the emphasis on data, the discussion of what controls are needed and why, and the times when really smart scientists disagree on some aspect of the paper, gives listeners an understanding of why science matters and how scientists think. It provides a lively, fun learning experience for us science nerds, together with the many, many non-scientists who enjoy listening to every podcast of TWiM. Happy anniversary, TWiM, and thanks so much to the TWiM team.
That's just absolutely incredible. And Elio, Stanley wrote to me after he sent in the video, and he said, parenthetically, I think it's time for Elio and me to update our article on the impact and future of microbiology. So you have a homework assignment.
Yeah. I got problems.
But he also added, he gave us some perspective in his note back. He was going to record another video, but he said he just sent me a couple of bullets. And so rather than reading all of them, I'll dump them into the show notes so that folks can look at it. But they all can be summed up by thinking about clever genetic approaches that will play a key role in discovering whether it be a narrow spectrum antibiotic, whether it be a new approach for environmental analysis using nanosensors. Then we need a deeper understanding of metabolism in physiology. And I know Amy Chang-Vollmer has always been a strong advocate of reinforcing the importance of teaching the undergraduate community the issues of microbial metabolism and how physiology is paramount in understanding our microbial world. And finally, that microbiology will play a foundational role in transdisciplinary team science required to solve the grand challenges. And it really is vintage Stan Malloy, really laying down the gauntlet for everyone to work towards.
Hear, hear.
All right. Our next guest is Vanessa Sperandio from UT Southwestern, University of Texas. And Vanessa was on TWiM 130, which we recorded in Boston at ASM Microbe. We were all there, Elio, Michele, and Michael.
Right.
And we had Vanessa and another guest, David Schneider. And then more recently, we've discussed one of Vanessa's lab papers on TWiM. So let's hear from Vanessa.
In this past year, I think some of the most exciting things that I've seen in science have to do with the marriage of biology and engineering, and how we took to the front lines organoids and basically putting this into nanoplatforms in which we can actually combine engineering and biology to actually study stem cells, making microorgans, be it like intestines or brains or lungs, to study infections. The other microbiology question that has been in the forefront and it's been exciting to follow is the mechanistic relationships between the microbiota and the hosts, and how mechanistically these relationships interact with each other, as well as with invading enteric pathogens. The other part that is really interesting to me is the gut-brain axis, and trying to get to the mechanism on how microbiota and also pathogens influence the host's brain and behaviors, and vice versa. So those are the things that have been really exciting to me in science in the past year or so. So that's it. Thank you very much.
Nicely put.
All right.
She emphasizes how, as we learn more about the interactions between the host and the microphysiology, it puts pressure on the experimentalists to come up with devised systems, experimental systems, to get in and ask those questions. And certainly she mentioned a number of the approaches that are really promising. And Vanessa, we're lucky to have you at the forefront of that effort.
All right. Our last guest is Marvin Whiteley from Georgia Tech. Yeah, Marvin was on TWiM 208, and that was when we visited Georgia Tech. Michele and Michael and I went there, and we recorded with Marvin and a couple of other faculty and postdocs. And Marvin is a big TWiM fan, so I thought it would be nice to hear his thoughts.
So I'd like to start off today by congratulating TWiM on 11 years. I'll just say, I really love TWiM. It's one of the things I really look forward to in microbiology, on the drive in from work, or I actually listen to it when I work out. I don't know what that says about me, but that's what I do. And please don't tell my virology colleagues like Chris Sullivan, but I actually listen to TWiV also and learn a lot in TWiV. So, the TWiM folks do a really great job. I also would, I think we should give a shout out to ASM, and ASM does really great things for microbiology, and I've become a real proponent of publishing in ASM journals and instead of other for-profit journals. So I think this sort of TWiM and ASM doing such a great job. And when Vincent sent me an email and asked me to sort of comment on how microbiology has changed in the last 10 years, one of the first things that actually came to mind for me was TWiM and social media outlets, right? Like Twitter, mainly Twitter for me. And, when I was in grad school in the late 90s, there weren't that many journals that we actually looked at as a grad student. We waited for bated breath for the sort of the physical copy of the Journal of Bacteriology to come to the lab, and we would flip through it. And also molecular microbiology was big in the field that I was in at the time, and we were always sort of flipping through these journals. But the students now and trainees now, there's just this explosion of journals, and you can basically get almost anything published somewhere now. And you have journals with 90% acceptance rates, and trying to find papers that you'd like to read. I think they find it hard. And places like TWiM and Twitter, do a really good job. I think Twitter, especially for me, is a place where I can go on and, in 5 or 10 minutes, sort of find papers that, it may have taken me a while to actually search for. So I think that social media aspect of just finding papers and finding science that's out there is one of the things that's changed quite a bit in microbiology in the last 10 years. Another thing that I think's changed a lot, certainly in my lab, I started my lab in 2002, so almost 20 years now. And I think it's computational biology. If you would have told me in 2002 when I started my lab that I would be, scouring the country looking for, people with math degrees that are interested in looking at big data sets and doing computational biology, I would have just thought you were nuts. I thought, I'm a molecular microbiologist. It's what I'll be doing probably for the rest of my life. But I think now, it's almost essential, at least I feel like, I personally believe that's essential, that computational biology be a part of almost any training now. Because even if you're a molecular microbiologist, I think there is value in actually going in and being able to download data sets and actually analyze them in a way so you can look at and put maybe just put a little bit of context around your around the project that you're working on. So I think that's something that's really changed, and I just don't really see that changing much at all. And I think it's just going to become more prevalent. And I think it's really sort of impacted the way we teach also, right? I teach undergraduate microbiologists, and, these folks they're really interested in me trying to tell them all the new cool things, right? And of course, COVID is something really big right now. They really want to understand COVID. And it's really easy to get caught, at least for me as a teacher, in the trap of trying to be cool and them liking me a lot and giving them all this new information without sort of giving them the basics. And so I think there's been a lot, at least for me, in teaching, trying to really understand, how can I actually get them to learn basic microbiology and understand microbial physiology and the things I think that they should really know, but still make it interesting and contextualize what's actually occurring now and give it some relevance to what we're actually working on. I guess the last thing I would say is, I think the last 10 years have been the most fun that I've ever had as a scientist. I would go back to grad school in a heartbeat. I told my wife this the other day. She's not super excited about that, if that were to happen. But I would go back now. It's just, I had so much fun in grad school, and I just can imagine how much fun it would be now with all the new technology and all the new knowledge that we have and sort of the new perspectives on microbiology, whether it be computational biology or whether it be evolutionary biology, which has really made inroads into microbiology. I think all of those things would be just so much fun to be in grad school now. And, so it's this multidisciplinary nature. And I think science was always, and microbiology was always multidisciplinary, at least since I started in the late 90s, it was multidisciplinary. But I think certainly this has just moved forward even further now. And so that's kind of my perspectives. I hope that's kind of helpful. And again, congratulations, TWiM. Keep going. I need something to work out. I need podcasts to work out too. So good luck.
Twice a month, whether it kills you or not, right, Marvin? Just kidding. So that's at Whiteley Lab, Marvin Whiteley on Twitter, for those of you who use that media.
I think his 2 points, I would agree on. Also, the social media has changed science. It's allowed people to interact way more than they ever did. And that's what we're doing here as well. But there's also other forms. And I think the computational aspects are incredible. Yeah. I think we've already talked about this. So I'm wondering if all of you have some thoughts about what do your impressions of the past 10 years. Let's start with you, Michele.
Well, yeah, I'll be brief. Our colleagues, what a great lineup you put together there, Vincent, emphasized a number of the advances in our field that I also admire. But I want to say that as I reflect on the last 10 years, what I appreciate is that contrary to the popular stereotype, microbes do not live a solitary existence. They live in communities. And that's the same for microbiologists. We do not sit in our labs staring down the microscope or looking at our culture. We actually thrive in a community where we have division of labor, we share resources, we are more resilient. And I am just really grateful for the amazing community of microbiologists that I've been a part of, certainly with TWiM, but back from my early days in graduate school. And I look forward to many more.
How about you, Elio?
Well, I find it interesting that several people emphasize this, how much microbiology has changed to include not just computational science, but also the concept of ecology. That is, microbiology has become an ecological science. And this is what actually happened about 1910, when the Dutch School of Ecology started. Vinogradsky and other people like that, they thought that this was an ecological science. Then what happened was molecular biology. Molecular biology took over, and that was it. Molecules, molecules, molecules. And now we go back to an early stage, and I welcome it very much. And I think that people were extraordinarily articulate in what they said. I'm very grateful to them.
And in fact, Elio, we go back to ecology, but now we have better tools, right?
Exactly. Now we can do it. That's right.
In fact, when I look back at all the papers we have done, some of the most interesting to me are ones where we explore the role of microbes in the environment and rocks on space or wherever at the bottom of the ocean, which is all new to me, and I love it. And I look forward to more of that. What about you, Michael?
I think that to quote Mark Martin, we have a quality quorum. We couldn't do TWiM without all of your hard work out there. The papers that you folks are publishing are effectively the topics that we hunt for each week as we're trying to assemble what we're going to talk about. And we couldn't do it without you. And, making your work accessible and helping us discover it, whether it be through Twitter or whether it be through the journals, enables us to do TWiM justice. And I think that's my impression is that we have a quality quorum. And I agree. Some of the most interesting topics that we have covered is the soil composition that makes the best wine. That was one of the topics out there. And, there's a whole emerging field of microbiology looking at, as some of my graduates would say, useful fermentation. And where you really exploit the microbes to make the finest beer and ale, and of course the wines that our friends in California are continuously researching, as well as France and other areas of the globe. So keep up the good work, folks.
One last comment. This social aspect of this new social aspect driven by the internet and so forth. What this does is when we go to do TWiM somewhere, people come up and they know us and they say hello. They're our friends, and this would never have happened before. Right. And I really enjoy that because you would know a few people in your field and that's it. So I really enjoy that.
Thank you, Vincent. We are all part of what you created. And your creation, you put together an unbelievable set of activities. Really, thank you.
Set the bar high.
Maybe the only thing I ever created.
You have two kids.
Three kids. Three kids. All right, that's TWiM 237. You can find the show notes at microbe.tv/TWiM. If you want to send us questions or comments, TWiM@microbe.tv. If you like what we do, consider supporting us. Microbe.tv/contribute. What is TWiM? TWiM is Michele Swanson from the University of Michigan. Thank you, Michele.
Thank you for 10 great years.
Elio Schaechter from Small Things Considered. Thank you, Elio.
Thank you. My goodness.
And Michael Schmidt from the Medical University of South Carolina. Thank you, Michael.
Thanks, Vincent. Here's to 10 more years.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.